screen. Okay. And we're at the end of section 2. No, end of section 3. So we're starting section 4 today. Okay. But what you are really going to need is this.
Great, like it makes sense, but I was like,
Well, that's why you kind of need.
okay.
We need a little. That's why we need a little reinforcement today. Recharge yourself by the wall.
It's fine. It's not gonna die on me, I don't think. But
Right, but it's always going to be on the same paper. The old school.
yeah. I thought that.
I read some kind of post. It was like, how do you know you're old? And there were all the things that I knew, and I was like, oh. Oh. It happened. But that's okay. Better than the alternative. Ready to need a pen? I also brought pens. Because I really don't want you to look at your notes. Turn the paper over. Don't read it yet. Don't read it yet. I want us all to kind of do it together. Don't look at it. Don't look at your notes when you're trying to answer it. Don't look at it yet. It's going to be okay. I'm going to teach you some ways, some things, some trickies. Okay. So if you're on Moodle only, go to the class, and then you're going to want to go to chapter 9, all the way at the bottom. Here, why is glycolysis great? That's what you're going to need. It is great. You're like, oh, not on Monday morning. So don't start answering yet. Just flip it over. We're going to do this together. How much time we got? A minute? 2 minutes? It's like as the semester goes on, there are so many fewer people in class. Imagine that, right?
There was 4 people in my English class today.
Do they Zoom?
No. No, there was just 4.
No, there was just 4 people.
We only have like 10 people in the class.
But still.
But there was 4 people. There was 3 parking well too. Like.
Oh, getting to the end, it gets tough.
I was tempted not to go, but I just can't skip class.
No, I was that way too. I just have to be there.
Yeah, because you know what? The one time you don't go,
There's like some pop quiz or some bonus something or some.
bonus points.
Yeah. But and y'all know y'all bonus points. What do you have to do to get your 5 bonus points on the final exam? 70%. I need to check. I haven't checked the percentages yet. I need to check. So hopefully by next class I'll check and I'll let y'all know. So 70% or above, get it. It's 5 bonus points on the exam. So y'all been watching the news? So did y'all hear they found a genetic link to diarrhea? Y'all heard that? Yeah, it runs in your genes.
Don't say.
It is.
It's a joke.
It runs in your genes. It runs
Oh
in
my
your genes. So I technically forgot Charles's joke for the day. So that's my joke.
I thought you were being serious.
Yeah, that was the plan. So did y'all hear that oxygen and potassium went out on a date? It was all right. It was okay. It's okay. All right, that's the end of my bad science puns for the day. All right, so if you just walked in, you should get a sheet of paper. Come get a sheet of paper. We're going to try this. Sorry, I missed some of y'all walking in. We're going to try this. Flip over your paper. Do not look at your notes. Close your notes. Don't look at anything. Don't look at anything. Have your sheet of paper. If you need a pen, I have some extra pens up here. But let's try to answer this. So why, oh why, is glycolysis great? It's not a trick question. It's not a trick question. What do we get out of glycolysis? We get ATP. So we get how many? 2 ATP, but these are not just any ATP. These are special ATP. Why are these special ATP? Because what? Well, it is a net of 2 ATP. That is true. We get 2 ATP. This is net. But why are they special? Did we have to shuttle them? Did we have to go all the way to the mitochondria? Do we need oxygen? No. So these are independent of the ATP synthase mechanism, which means you have to go through citrate, right, to the electron transport chain, to ATP synthase, right? So that's a big, long pathway that we had to take to get to tons of ATP, which is great if you have oxygen. But what if you don't have oxygen? You can't do it. So the only ATP you can generate if you don't have oxygen are the ones in glycolysis. Okay, and later today we're going to talk about what happens in those cells that don't have oxygen. Like, okay, glycolysis happens, but what else happens? And so we'll talk about that later. What else? That's part of it. Part of answer number 1. We get 2 ATP, and those 2 ATP are independent of ATP synthase. But what else? So that's 1 point. What else did we do in glycolysis? That very first step. What's that very first step? We go from glucose to glucose 6-phosphate. This is awesome. Why is this awesome? Don't look crazy. It can't leave the cell. This is the big thing. That now we have trapped our substrates for ATP synthesis. Right? We've made them negative by making them negative, right? By making them negative. Right? So we made them negative. That's amazing. And it's amazing because now they're trapped. They can't just diffuse out, because that's how we said glucose gets in. It's some diffusion mechanism. Sometimes it's facilitated, sometimes it is not, but we want to trap it and we want to make sure that it doesn't get out. Okay. What else? Anything else? That's the 2 big ones that I have. But you know, you might have some additional things to add. Like, no. Okay, the second one. Number 2. Do not look at anything. Do not look at anything. I want you to try scroll up. What are the investment and payout reactions in glycolysis? Give the substrates, enzymes, and products, and indicate ATP lost or gained. I don't care if you only get 1. I'm not grading this, but I don't want you to look. Take a minute. I'm going to be quiet and try. gave you part of one already. Hint, hint. Which one? They were both so amazing today. What, say it again? The jokes? It ruins it, but it's okay. It ruins them both, but it's okay. It's okay. I'll tell you after. Okay. Talk to other people around you. See if somebody came up with something you didn't. Just turn and talk. See what y'all came up with. I have a message in the chat.
Okay.
I'm sorry. What is the word after making and before negative in the second part of one? By making them negative. Them. T-H-E-M. Them. Them. Yeah. Them. Thank you. And y'all talk. Talk. Okay, everybody should have the first one. What's the first one? The first one's reaction 1, right? What's reaction 1? Glucose, GLC, the abbreviation for glucose is GLC, plus what? ATP yields glucose 6-phosphate. Okay, did we gain or lose? So this is a negative amount of ATP, one loss of ATP. Enzyme, hexakinase, or glucokinase, but glucokinase is specific, and we're going to talk about that in a little bit, but it is only in liver and pancreatic cells. Okay, so this is really our investment, right? So like step 1, that's what we kind of called it in the notes, right? Okay, there's another step in the investment phase, right? Remember, we had to invest 2 ATP. So we're going from fructose 6-phosphate, and we're going to make 1,6-bis phosphate. That's right. Very nice. What is common in our 2 investment reactions? We're losing an ATP in the cell, and we're putting that phosphate group onto a substrate. First it's glucose, then it's fructose. Right? Yeah. Okay. So what's the enzyme? Phospho, P-H-O-S-P, phosphofructokinase 1. A lot of times it's abbreviated as PFK1.
So do you put like minus ATP?
You do. You do. So this is a loss of 1 ATP. Thank you. Thank you. Okay, so that's reaction 3. If you're looking in your notes and you want to like go back to that, they're all numbered, and that's number 3. Okay, how much ATP do we have to invest? 2. We've invested 2, so now we're really in the payout, which is like step 2. It's not really step. Is it, do I list it as phase? Do I say step? I don't know what I say in the notes. Step 2, right? Phase 2, whatever. Oh, I gotta extend. I'm scrolling down and nothing's happening. Okay, so now what are we gonna be doing? We're gonna go the other way, right? We're gonna pull phosphates off and make ATP, because how many ATP do we need to make? 4. So we're gonna make 2 and 2, right? So we'll just do that, plus 2 ATP plus 2 ATP, and these reactions are going to give us, well, actually, you know what? It's actually not plus 2 ATP. It's actually this whole reaction times 2, which gives us 2 ATP. That's a better way of saying it, right? So let's do that. Times 2, we're going to make 2, move this down, 2 ATP. Okay. So what do we start out with in this next phase where we're going to, not plus ATP, yeah. Oh, I'm counting that as a product. Yeah, it's gonna make, okay. I was like, what was I thinking? I just didn't put my arrow. All right, so what do we start with? What are we gonna phosphorylate? Or dephosphorylate, actually. What are we gonna dephosphorylate? We're gonna have 2 phosphate groups on it, right? So it's 1, 3. What does BPG stand for? You can go back and look at your notes now. It's okay. Bis phosphoglycerate. And we're going to remove one of those phosphate groups. And what are we going to get? Just phosphoglycerate. Did we leave it on the 1 or the 3? We left it on the 3. So what kind of enzyme? It's a, what's the similar? Phospho, PHOSPH, phosphoglycerate, CER. And what are we doing? We're phosphorylating, in this case, ATP. So it is a kinase. Because really, what are these reactions? They're all technically reversible, technically. But we don't call them that way because of their Gibbs free energy, right? Okay. Yeah, this one would be reversible. This one really is because it has a delta G close to 0. Yeah. Okay, now what? Which, that was number 7. And the last one is the last. Look how you're starting. You start with the first and you end with the last. Yeah, okay. So now what are we starting with? What do we end with? Let's do that. What do we end with? What's our product? Pyruvate. Okay, so that's an easier way to do it. So if you're going to end with pyruvate and you're going to pull a phosphate off, you got to start with phospho something pyruvate, phosphoenol, SP phosphoenol pyruvate. It's really one word, but I didn't leave myself enough room. So if you had phosphoglycerate kinase above, what do you think this one is? Name the product. What's the product? Pyruvate kinase. Not so bad, right? Why wouldn't it? No, because you're using the name of the product.
No, I'm not talking about the enzyme. I'm talking about like pyruvate itself. Because like it's phosphoenol pyruvate.
It's only an enol because you have a phosphate on it. No, it's okay. All right, so we got these 2 ATP. These are plus, what's our net? 4? No, because we lost 2 up here. So we have a net of positive 2 ATP. Yeah? Okay, so these are all of our investment and payout. That was the question, right? What are the investment and payout reactions in glycolysis? We gave the substrates, gave the enzymes, gave the products, and we indicated ATP lost or gained. Awesome. Good job, y'all. Don't you feel so proud? Yeah. Okay, now, here's a tough one. Now that you have that one answered, here's a tough one. What are the irreversible reactions in glycolysis? So all I'm going to do is I'm going to change this to number 3. Is my answer, right? Is reaction 1 reversible? No, it's irreversible. Okay, so I can leave that one. Is reaction 3 reversible or irreversible? Irreversible. Reaction 7 reversible or irreversible? This one's reversible. So what I can actually do is just scratch that one out. My last one, reversible or irreversible? Irreversible. Oh, seeing some overlap, aren't we? Interesting. Interesting. So we answered number 3, right? Gosh, that must have been the quickest we've ever answered anything. So what do you notice about, I'm going to ask you 2 things. What do you notice about the names of the enzymes that catalyze the investment and payout reactions? They're all kinases. Does that make sense for investment and payout? What do you do in an investment and payout? Shuffling around phosphates, right? What do you do that with? Kinases. That's a, like when you're taking a test, especially if I have some multiple choice questions that are like, pick the enzyme that does whatever it is. Look at the names and draw some conclusions from that, okay? It's a really good test taking skill. All right, last one, right? What factors control flux through metabolic pathways such as glycolysis? Okay, let me scroll down. Maybe I just need to go to a new page. Okay, new page. So what controls flux, metabolic flux? Well, what controls a metabolic pathway? Enzymes, right? So enzymes control metabolic pathways. So the question is, what controls the metabolic pathway? Well, whatever controls enzymes. So what controls enzymes? And y'all said it. Enzymes controlled by, y'all said it. 2 things. Substrate. We said this first. Substrate. What? Substrate concentration. Which reactions are controlled by substrate concentrations? Are all of them? Concentrations control reversible reactions. So how do I know something is reversible? It has a delta G close to what? Close to 0. So tell me about these enzymes. How are these enzymes working? Like, how catalytically active are these enzymes? Do we want to control their catalytic activity? Why? We're already controlling the reaction by substrate concentration. Do I need to control the enzymatic activity? No. I want them running at full speed all the time. That way I can switch back and forth just on substrate concentration. See what I mean? So that you have an instantaneous result just based on substrate concentration. So these enzymes are working, that's working at full capacity. So it's all about substrate availability for these reversible reactions. Okay, what else are enzymes controlled by? Catalytic activity efficiency. What kinds of enzymes in glycolysis are regulated through their catalytic activity? Reversible or irreversible? Irreversible reactions. this so important? Let me stop you, and I want you to think big picture, okay? Let's think about us as cave people, not in our normal today setting, right? Let's think about cave people, right? They're hunting, right? And they make a kill, they eat the food, right? What if they eat more than their body needs at the moment? We want to store it because winter's coming, right? Winter is coming. Isn't that what they say? Winter's coming. Yeah, okay. So we need to be prepared for that. Our bodies need to be prepared for that. So instead of going through glycolysis, I might want to use those sugars to make glycogen, to make fats, to make backbones for amino acids. Do you remember the forest? Right? Sugar comes in and we're picking like one tree in the forest. There are lots of other places that sugar can go. And so you have to kind of think about that. So I need to have a way to regulate the whole metabolic pathway, even if my substrate level concentration is high. And the way that I do that is through regulating catalytic activity. So how do I know that these are the reactions that are going to be regulated by catalytic activity? Because these are ones that the enzymes aren't always working at full capacity, right? So these enzymes can be regulated independently of substrate concentration. So that means you can have low enzyme activity with high substrate concentration, right? That's the definition. How do I recognize them when I'm looking at these reactions biochemically? They have what? Large negative delta G value. Okay, so does that help you kind of integrate what we did last time? It was kind of a lot, but what I want you to do is I want you to be thinking about it and thinking about what it means and all that fun stuff. Because glycolysis is awesome. Which leads us perfectly, absolutely perfectly, into section 4, right? So if we look at our glycolysis pathway, right? Do you remember the little animation they did of this? And when you went from glucose to glucose 6-phosphate, it went slow, but every step from glucose 6-phosphate to fructose 6-phosphate, it went fast. Then it was slow, and then it went fast. Do you remember that? So the reason why it's going slow, they're showing it as a bottleneck because these enzymes can all be regulated. How do we regulate enzyme activity? Post-translational modifications like phosphorylation and how do we regulate? That's the question. So allosteric binding of regulatory molecules. Should I write that? Okay, so how are enzymes regulated? We have post-translational modifications. And we have allosteric regulation by molecule binding. Right? You have some molecule that binds. Is it always a negative? Is it always an inhibitory effect? No, it can be something that will promote the reaction. Okay, so these enzymes are going to be enzymes that are regulated. So what we're going to do is we're going to pick one of these enzymes and we're going to talk about how we regulate one of these enzymes. And the enzyme that we're going to look at is PFK1, phosphofructokinase 1. All right. So this is one of the enzymes that will control the flux. Is it the only enzyme? No, there are how many? 3. Okay, but we're only going to look at one. So let's think about this. When do we want glycolysis to happen in the cell? Low energy, right? I haven't eaten for a while. I haven't taken in glucose. I have a low energy, which means I don't have a lot of ATP. But what do I end up having a lot of by definition? ADP and AMP, right? So both ADP and AMP are positive. I should not have done that in red. These are all positive influencers of phosphofructokinase 1 activity, right? They will promote the catalytic activity of phosphofructokinase 1. Fructose 2,6-bisphosphate comes in from another pathway, and you don't have to know that pathway. So I don't care so much that you know that, right? And now the opposite is true, right? If we have ADP and AMP positively regulating phosphofructokinase 1, then we want ATP to what? Negatively regulate. So that's true. So if you think about it, ATP is at the end of the pathway, right? We go glycolysis, citrate, electron transport. Then we make ATP. Couldn't we have an inhibitor that would be closer up in the pathway? What's the next step after glycolysis? Just said them. Citrate cycle. One of the initial substrates in the citrate cycle is citrate. That's why they call it citrate cycle. Imagine that. So if citrate cycle stops, what happens is citrate builds up. So if you inhibit the citrate cycle because you've made ATP, then citrate's going to build up. That built up citrate is going to inhibit glycolysis. So every step along the way is inhibited by the end product, and then each step along the way inhibits the first one. Okay, so glycolysis is our first like major pathway, right? Then citrate, then electron transport chain, and ATP synthase. So if you make lots of ATP, you're going to inhibit the citrate cycle because you don't need to go through citrate if you've got ATP. Okay, well, also ATP inhibits glycolysis. So the end product of ATP inhibits both citrate and glycolysis. When the citrate cycle gets backed up because of lots of ATP, you build up citrate. Citrate will go and inhibit glycolysis. So the end product stops up everything else. And if you inhibit one of the pathways in the middle, that will go and inhibit a previous pathway. Yeah? But it makes sense. Do you want to futilely make ATP when you've got tons of ATP? No, that's a waste of energy. Winter is coming. You need to store that. Okay. All right. So, yeah. So the how do these things, any of these things, regulate PFK1? And I just like drew a line right through PFK1. But how do these things regulate it? We said they're positive and they're negative. Are they going to phosphorylate it? Are they going to do a post-translational modification? No, we don't want it to be that permanent. We want it to be on and off really quickly. So what's an easier way to have that on-off switch? Allosteric modification. Right? So what does that mean? When this inhibitor of ATP binds to phosphofructokinase 1, does it bind at the catalytic site? No, it binds at a site other than the catalytic site, induces a conformational change, which then inhibits the activity. The opposite is true. So that's where we say, okay, we added ATP, right? That's here, that's in the red. We add ATP, what's gonna happen? It's gonna be inhibited, right? Now, what if we add a DP? It's gonna go faster. You could add other activators and it goes even faster. But it's just allosteric regulation. And so the activators will convert the conformation of the enzyme such that it has a really high catalytic efficiency, right? Whereas your inhibitor is going to give it a low catalytic efficiency. So if it's in the inactive state, here's inactive. The inactive state is the T state. The active state is the R state. Remember we talked about hemoglobin? Remember that? T state and R state? R state, you had oxygen bound. T state, it was unbound. It's the same sort of deal. It's the same sort of deal. Okay, so do y'all remember when I was talking about glucokinase being a glucose sensor in the cell? We were talking about testing your blood sugar. And how does your body test your blood sugar? Your body tests your blood sugar with glucokinase. Where is glucokinase found? Pancreas and liver. But today we're going to talk about the pancreas. Okay, so we're in the pancreas. Why would we want, what's the difference between glucokinase and hexakinase? Let's talk about that real quick. Glucokinase versus hexakinase. It is, but I specifically want to talk about the reaction from glucose turning into glucose, GLU, glucose 6-phosphate. It's true. But in this reaction, we said it could be either one of these enzymes that makes this reaction happen, right? You need ATP regardless, but how is it different? Hexakinase is in all of our cells, right? This one's the one in pancreas, liver. So glucokinase. Do you remember how it's different? There are two things. It is not inhibited by things like ATP. Or what else? Do you remember the products inhibit the reaction, but the products end up or glucose 6-phosphate, right? Because if glucose 6-phosphate builds up in most of our normal cells, that means that, golly, we've made a lot of ATP. We don't really need all this glucose to go to making ATP. So we want the product to inhibit that initial reaction so that we don't go through things that we don't need to. Okay, so it's not inhibited. That's a big one. What else? It has a low binding affinity. For glucose. Now let's think about why that's really important. If I have a little bitty snack, right? If I have a little bitty snack, let me see. This is going to be my blood sugar levels. My blood sugar levels. My BS level, right? Okay. This is where I kind of want to be. This is like the happy place, right? Normally, what does your blood sugar look like, right? If I eat a meal, it'll go up. I'll make insulin. Insulin will tell all the cells, hey, take in the glucose, my blood sugar will drop, right? And it cycles like this throughout the day. Now, if you like drink a 2 liter of Coke, you're going to have something like that, right? It's really bad. But what would happen? How would my graph look different if I had a high binding affinity for glucose by glucokinase. What? It wouldn't always be high. It would be every little bit of glucose would trigger my pancreas to secrete insulin, because that's what your pancreas does, right? Secrete insulin. So it would look something like this. That's not efficient. That's not good for your body. It's not. Okay, so what's going to happen, right? If we have a low binding affinity of glucokinase for glucose, it's going to take us eating enough to rise, to cause a high enough rise in our blood sugar to activate glucokinase. Now the glucokinase is active, I don't want to shut it off right away, right? So I don't want to inhibit it. I want it to go through its pathway to make insulin, right? So this is kind of what I was saying before on how glucokinase stimulates insulin production, right? We go through our normal glycolysis, right? What do we make? We make pyruvate. Our pyruvate goes through citrate, goes through electron transport chain. We eventually get lots of ATP being made. The high level of ATP is going to inhibit a potassium channel. When that potassium channel is inhibited, we get membrane depolarization. And that means that we have these calcium ions that are normally sitting outside the cell that get pumped in. And we know that ion concentration differences can induce fusion of vesicles in cells. This is how our neurotransmitters are released in our synapses, right? We talked about that before. It's the same sort of deal, except in these vesicles, we have the hormone insulin. And so that's what triggers insulin release, right? So we don't want glucokinase to be shut off too early. We don't want it inhibited. Otherwise, we'd release a little insulin and then it would automatically shut off. And our cells wouldn't get enough glucose. All of our other cells wouldn't get enough glucose, right? Cool? Okay. So you got to kind of think about the big picture. So when we think about glucose, we've only been looking at kind of this blue box. You see this blue box in the background, right? We go from glucose to pyruvate. And there are lots of different ways that we can get substrates into the glycolytic pathway. We can start at the beginning. That's an okay thing to do. Any kind of plant starch is metabolized into maltose, right? And maltose is just multiple glucose units hooked together. That's maltose. You could also get it from your diet. Where do we get lactose? Milk products, right? And what is lactose? And you can look in this diagram and figure out what lactose is. What are the two things that lactose turns into? Glucose and galactose, okay? Now, the glucose that comes off of that lactose can go straight into glycolysis. That's awesome. But galactose is not a substrate for glycolysis. Remember, we said that enzymes are super specific for their substrates. So they have to be converted. And one of the enzymes that's required for that is galactokinase. And we're going to talk about that in the next slide on exactly how that works. But eventually you can get it converted to glucose 1-phosphate, and then it can enter glycolysis. Sucrose. What is sucrose? Table sugar, right? Table sugar is a fructose and a glucose, right? So whatever you have in your cabinet is sucrose. That's table sugar. So the glucose can go straight in, but fructose has to be converted. Inside of muscle cells have hexakinase that can convert it straight from fructose to fructose 6-phosphate, right? And then we can enter glycolysis. You could convert it to fructose 1-phosphate and then go through some intermediates to eventually get glyceraldehyde 3-phosphate. But do realize that if you start with fructose over here, you're only going to make one molecule of glyceraldehyde 3-phosphate. So how many ATP are you actually going to get out if you start with just fructose? Only one, right? So it all depends on where they enter the glycolytic cycle, how much ATP you're actually going to get out of them, right? But what I want you to realize is that they're going to come from other sources. You can metabolize fats. Fats are normally found as triglycerides inside the body, right? We have lipases that will cleave the fatty acid groups, right? A triglyceride. How many fatty acids do you have hooked to a triglyceride? 3. So we can get our 3 fatty acids. We can convert those to acetyl-CoA. And once we have acetyl-CoA, what can we go through? Citrate cycle. Generate lots of ATP, right? We also end up with a glycerol molecule, right? A triglyceride means that we have 3 fatty acids in a glycerol. So our glycerol can be converted into glyceraldehyde 3-phosphate. And that can enter the glycolytic pathway, right? But you only get one of them that way, right? But then you're getting another ATP from the fatty acids. But you just got to realize that it can come from multiple different sources. You don't have to particularly eat glucose, right? We typically don't do that. We typically eat something else and then metabolize it. So here's kind of our example for galactose metabolism, right? So you start out with galactose. And what you have to do first is you have to phosphorylate that galactose molecule. Now, here's the deal. Our bodies can't really convert galactose straight to glucose. But what you can make is UDP glucose. So this is a glucose molecule that has, what is this? A P, a ribose sugar, and a base. Do you see how this is UDP? Diphosphate. Yeah. So uracil, a ribose sugar, and 2 phosphates. Yeah. And then we attached what to it? Glucose, right? So what we can do is basically exchange. What's the difference between glucose? This is a glucose molecule. This is GLC, glucose. This one is galactose. What's the difference between galactose and glucose? Okay, so that's carbon 1, 2, 3, 4. On carbon 4, we have a hydrogen and a hydroxyl that are swapped. What do we call those? Epimers. Remember epimers? Don't forget to study that stuff. So we can't directly convert it, but what we can do is swap this galactose for this glucose. So now we have a free glucose, and the galactose is now hooked to our nucleotide diphosphate, right? So our glucose 1-phosphate can then go on to glucose 6-phosphate and be metabolized in the glycolytic pathway. But you have to have an enzyme that can convert this UDP galactose back to UDP glucose. And we do have an enzyme that can do that. So when people have, you know how I've talked about people have lactose intolerance, right? They're missing the enzyme that allows them to hydrolyze the lactose, right? You can have a galactose intolerance. And it's actually more severe because you cannot convert this back into UDP glucose. And so you actually end up building up tons of this UDP galactose, and it actually becomes toxic to the person. And they have pretty severe symptoms. But you can treat it just by going on a galactose-free diet, right? But a lot of times you see it in little kids and stuff like that. Okay, so the other thing that you kind of have to think about with glycolysis is that glycolysis doesn't just provide ATP. It also provides the carbon backbone that's required for lots of different metabolites inside of the cell. So for example, if I would like to make nucleotides, right, I can go through the pentose phosphate pathway and I can actually make backbones for nucleotides. If I want to go and store glycogen, right, I've been working out and used up the glycogen in my muscles. I need to rebuild my muscular glycogen storage or liver glycogen, doesn't matter, right? remember, winter is coming. So if I don't need to make ATP right away, what can I go and make? Triglycerides. Do you see how in the first slide where we kind of looked at this kind of a diagram, everything was feeding into glycolysis? This diagram is the opposite. This diagram is everything going out of glycolysis. It is not just the end product of pyruvate to go through citrate to go through electron transport chain, right? You need those carbon backbones for lots of different things. What are serine, tyrosine, alanine, aspartate? These are amino acids. Amino acids. So you're going to make amino acids. That carbon skeleton is super important for lots and lots and lots of different things. So don't think about it as only a tree, right? Those trees are all connected in the forest. All right, so we've made pyruvate. Now we're back to the tree, right? What was our product? How many pyruvate did we make? 2. Pyruvate's metabolized in one of 3 ways, and it all depends on what organism you're in and how much oxygen is available, okay? So if we are under aerobic conditions, what does aerobic mean? Oxygen's present, right? We can go on to ATP production. This is typically what happens in most of our cells. We go through the citrate cycle, we go through the electron transport chain. What they don't say is we go through ATP synthase and we make ATP. What else do we get out of as an end product? Do you remember the very beginning we talked about cellular respiration? What else do you get? CO2, water, CO2 and water. Okay, so you also have the possibility for anaerobic conditions, right? So under anaerobic conditions, what's that lactate going to do? What's that pyruvate going to do? It's going to turn into either lactate or ethanol. And that depends on what organism we are in, right? So here's kind of a represented diagram. Here's our anaerobic metabolism, right? Because where are we if we are anaerobic? Anaerobic. We're gonna occur in the cytosol. We have no oxygen present, right? Okay, so now we can go into the mitochondria with our pyruvate. If we go through our mitochondria, we make ATP, right? Here, ATP, CO2, and water. But where might you be anaerobic in us and make lactate? In your muscle cells, in your red blood cells that don't have mitochondria. So the whole point of being able to convert pyruvate to lactate is really, really, really important. What enzyme did we purify in lab last time? Lactate dehydrogenase. Oh, imagine that, right? What does lactate dehydrogenase do for us? We make lactate, but that's not the really important bit. What's the important bit? When I went through glycolysis, in order to make my 2-ATP, I had to use up an NAD+ to make NADH. If that's it, if I don't make any more NAD+, glycolysis will shut down. I must regenerate NAD+. Super important. So lactate dehydrogenase will do that for me. Now we have some organisms like Saccharomyces Cerevisiae, right? What is that? Yeast, Brewer's yeast, that will convert the pyruvate to ethanol. Is ethanol super important? No. What's important? That fermentation process regenerated NAD+. We enjoy it, right? We enjoy beer, But that's not the important part. The important part is that the cell can continue to survive because we've regenerated NAD+. So, hence the next slide, NAD+ regeneration, right? Super important. So it's required in order to maintain our flux. So that step that requires NAD+ is the conversion from glyceraldehyde 3-phosphate to 1,3-bis-phosphoglycerate. That enzyme that catalyzes that is glyceraldehyde 3-phosphate dehydrogenase, or we abbreviate it GAPD or GAPDH, depends on who you want to say, right? But all of that happens inside of the cytoplasm. So if you have things like red blood cells that don't have a mitochondria, they can go ahead and regenerate that NAD+, and it's okay. Now, if you end up taking that pyruvate instead and sending it into the mitochondria to go through the citrate cycle and all that kind of stuff, you have some very, very important mitochondrial shuttle systems that will shuttle NADH, NAD+, back out into the cytosol so that you can maintain that flux. But there are shuttle systems that make that happen. Okay. All right, so what happens if you have a lactate dehydrogenase deficiency? What if your enzyme is not functional? What are you not going to make? You're not going to make enough NAD+, right? What happens if you don't make enough NAD+? You're what? Yeah, the pathway is going to shut down. You're not going to be able to make ATP. So people who have a lactate dehydrogenase deficiency, when are we going to see that in those patients? When what? Well, what are some situations where you might need high levels of ATP? When you're exercising, when you're working out, when you're running. So these people fatigue very easily, right? You can actually cause extensive damage if you continue to try and it continues to happen. But this is how patients get diagnosed with a lactate dehydrogenase deficiency, is that when they exercise, they fatigue very quickly. They'll recoup it, right? But then it's just those high ATP needed levels where you go without oxygen, right? That's the problem. When you can go through the mitochondria, then you don't see that issue, right? Okay, so here's the other thing that's really cool, right? If we don't take our pyruvate through the mitochondria, if we instead go through fermentation. So this is Brewer's yeast. I used to work with Saccharomyces Cerevisiae, and the lab either smelled like we were baking bread or brewing beer. It always smelled great. I loved it. I loved it. So how does this work? This is kind of an interesting thing that you don't necessarily think about if you're a beer lover, right? So what happens to make beer? Well, you have to start with some kind of a sugar, right? So you have a grain. And so what they do is they germinate the grains. What does germination mean? Anybody? Yeah, like start sprouting, like start, right. So when they start sprouting and all that kind of stuff, you have expression of the cereal grains, right? So that's going to be like our carbon source, and it induces expression of enzymes. So if we take that grain and we mash it, right? What we're going to get out are enzymes that are going to allow breakdown of those sugars. So things like alpha amylase and maltase. Remember I said maltose is the sugar that comes from starches, plant starches, right? So maltase is the enzyme that breaks down maltose, right? Okay. So if you add water to it and you heat it, What does heat do to enzymes? As long as you don't go too high. Increases reaction rates, right? So we start breaking down those sugars, right? So then what they do is they boil this, they call this at this point the wort, and they add other things in to make it taste good, like if you like beer with hops, right? So this is very beer specific, right? What they do. Now, this is the point where they add the yeast. So this is Saccharomyces Cerevisiae, right? You first incubate the yeast with oxygen. Why do we want to do that in the beginning? Because we want to give these yeast like a jump start, right? They can really grow. They can really be active. Then we automatically switch them to anaerobic conditions, right? Now they don't have oxygen. So now they're going to ferment all of that lactate into, all that pyruvate into alcohol, right? And so what you'll see is then they'll filter, carbonate it, and all that kind of stuff. Now, if you have like somebody, if you know somebody who brews beer on their own, what they'll do is right before they bottle it, they'll add an extra little bit of sugar and yeast, and it'll create that carbon dioxide that will carbonate on a small scale, right? So that's the difference between like a microbrewery and like a big production brewery, right? They do different carbonation techniques. All right, that's all I got for today. Hope that helps. Go study.
On Wednesday, I have presented my research at the national meeting of the ACS,
Yeah.
and that falls from 10:30 to 12:30, so I'm probably not gonna miss class.
So send me an email. I always record at Zoom. I'll send it to you.
Okay, that works.
Yeah, absolutely. Congratulations.
Thank you.
That's awesome.
Yeah, so-
The more experience you can do with that, the better.
Yeah.
I mean, that's awesome.